Histology Q&A session. My name is Wendy Xu, and today I'm joined by Dr. Yang, the first author of the article, Bladder Cancer Variants Share Aggressive Features Including a CA1 plus cell state and targetable TM4SF1 expression. Dr. Yang, thank you for joining us today.
Thank you so much for having me.
This study is about similarities in gene expression patterns for histological variants of bladder cancer. Dr. Yang, can you give us some background on what histological variants of bladder cancer are, especially with regards to your research, and what current treatments are for it?
Yeah, absolutely. So, I know it's a mouthful. The title's a mouthful, but simply, the histologic variants, they're basically bladder cancers. If you look at them when you're removing them surgically, they look just like any other bladder cancer. It's not until you look at it under the microscope that you realize that this is a cancer that is kind of mimicking something else. It could look like intestine, it could look like skin, it can even look like cells from the immune system. So these cancers are really, really aggressive. And before our paper, or before we did this project, there was very little that was known about them on the molecular perspective.
Yeah, okay. Thank you for your explanation. That's really interesting. My next question to you is, in your study, you used single-cell RNA sequencing. What would you say are the advantages of this technique in your study, and how do you see it being used to study other rare cancers or diseases?
Right. So, single-cell RNA sequencing, it's been emerging for the last decade or so. Essentially, what you're doing is you're trying to understand what every individual cell within a sample that you're analyzing, what it's doing at any given point. So what genes is that cell expressing? And you can even do things like look at the DNA, you can look at the proteins, you can even look at the metabolism. For our study, we focused on RNA sequencing, which is basically what genes were these cells expressing. And this is a really powerful technology used for, as you said, rare cancers, right? These histologic variants are quite rare. It's really hard to get thousands and thousands of these samples to do traditional analyses. So using single-cell RNA sequencing allowed us to extract as much insight as possible from a limited number of samples.
That's very fascinating. Thank you. And lastly, how do you see the results of your study translating to clinical treatments or becoming relevant to other scientific areas of research?
Yeah, that's a great question. And so the goal of what we were doing with this project was to find generalizable insights into these cancers, right? We knew kind of going into it that yes, each cancer, especially with these variants, is going to be very unique. But what we wanted to know with our big data set that we got from these limited samples, is there something that could be used not just for all the variants within bladder cancer, but also cancers in a broader sense? And so the two insights that we found were that variant cancers, even though that individually they're very different, express a protein called CA125 that can potentially be used as a marker to monitor cancer growth and progression and spread. So that was the first insight, and that was significant because we saw that in a number of different variants. The second major insight was that there is another surface marker called TM4SF1. It's a protein that is expressed in numerous cancers, and we developed a CAR T technology to try to target this cell surface marker and kill those cells, and we found that in a mouse model that we were able to do so.
That's wonderful. Very exciting work you're doing. Again, thank you, Dr. Yang, for taking the time to join me today.
Me too.